Good morning, everyone. My name is Michael Elzer. I'm the product manager at Takara Bio Europe, and I will present you today gene editing of iPS cells. And more specifically, I will show you an optimized culture system that supports your cells during the genome editing. So you will be muted during the webinar. If you have any questions, you can type them in the questions box, and I will try to answer them at the end of the webinar. So why would you want to gene edit stem cells or iPS cells? I mean, iPS cells by themselves are already very useful tools in cell biology, mainly because they can expand in a very robust fashion. So they have a good capacity to expand, and they also have a high differentiation potential, meaning that you can differentiate them into many different cell types. So that's already a very useful tool, especially for studying diseases. But if you combine iPS cells with genome editing, it allows you to study how a specific genetic modification can influence the function of the cell or an organ. More specifically, what it allows you to do as well is to create isogenic cell lines from healthy or sick persons. So what do I mean by that? So you can take either a cell from a healthy person, reprogram it into an iPS cell, and then do genome editing to insert a specific mutation, and then differentiate both of those cells, the healthy one and the mutated one, into your cell type of interest. And these two cells, since they are coming from the same donor, they will be isogenic, so they will have exactly the same genetic background except for the mutation that you have introduced. So in terms of control, this is a perfect situation because you can control, you can eliminate a lot of unwanted side effects by having isogenic lines. So they come from the same patient, they will behave exactly the same except for whatever the mutation is causing in the cell. Now we can do the same thing as well with cells from a sick person. So it's kind of the reverse process, but it depends really on the disease that you are studying. So here you will take cells from the sick person, reprogram them again into iPS cells, but then you correct the cells using a mutation. And also here you end up with two isogenic lines that can be differentiated into the cell type that you're interested in. And again, this gives you very good control for your experiment for many different downstream applications, like studying a new treatment for a disease, for example. So in today's talk, really, I want to start with a brief introduction to our culture system, which is called the Cellatis DefCS culture system. Then we will look at the gene editing part. First an introduction on gene editing, then why it is important to do single-cell cloning, and in the end I will show you an actual gene editing experiment using the culture system, and then we will come to the conclusions. So what is the DefCS culture system? The DefCS culture system is a complete and robust culture system for undifferentiated HPSC, so human pluripotent stem cells. So that's iPS cells, human iPS cells, and human ES cells. Now, complete in the sense here means that it's composed of the basal medium, the growth factors, but also, and this is quite special, it also contains the coating compound, so the substrate or the matrix that the cells can grow on. It's also feeder and serum free, so this prevents any problems from foreign materials, and very specially DefCS can be used for non-colony culture. So you can culture the cells as a monolayer, and this allows you to work with the cells like you would do with many other cell types, non-stem cells, meaning that you can do single-cell passaging and seeding. So this greatly facilitates working with stem cells. You don't have to do colony formation, dissection, or anything. You don't have aggregates. You just have a nice looking monolayer. And I have some pictures here what this looks like just after seeding. So these are iPS cells in DefCS. And you can see that we have a monolayer. We don't have really these clumps, and the cells they look very nice, spread out, but they don't, or they might not look like stem cells if you compare them to other stem cell media. But if you look after a few days in culture, when it's more confluent, you can see that they have the typical stem cell phenotype with a high nucleus to cytoplasm ratio, defined borders, and very prominent nucleoli. So this gives you several benefits. It's easy to count the cells and keep track of the growth. You will also be sure when you start differentiation that all the cells are at the same stage, so very homogeneous. They have the same environment. You don't have any colony edges, and this gives you also very good reproducibility for your experiments. So DevCS really supports human pluripotent stem cells for long-term maintenance. So if you keep your cells for a long time, also during the reprogramming, which is when you make iPS cells, and this is what we will talk about today, doing the genetic engineering and during the single-cell cloning. Now in the next slide I have some more data on the DevCS. So we have tested it with many different pluripotent stem cell lines, more than 85. It's very adaptable, so there are high chances that it will work also with your cells. More than 97.5% of the cells that we tested have grown positively in DevCS. The cell growth itself is very robust and has a constant generation time, so this also facilitates the planning of your experiment. You can really calculate how many days it takes. We have, and that's very important, a stable carrier type. We tested up to 30 passages. So in most cases, we don't have any problems with the carrier type, and that's also a very big issue with other culture media, so a big advantage. And of course, DevCS preserves the stem nest so that your stem cells will not do any unwanted differentiation, and we test this with different markers like OCT4, NANOC, SSEF4, and TRY160, TRY181. So you can see some pictures here. Okay, so that's the introduction for DevCS. So let's move on to the gene editing, just to look quickly at what steps are necessary to genome edit cells. So the 4 steps that are necessary and that I will talk about is, first of all, you have to create the correct editing machinery, then you have to deliver it to the cells, and when this is achieved, it's very important to create clonal cell lines, because we don't want to really work with a pool of cells that have different modifications or are even unmodified. This would really complicate things. And then in the end, we also have to do some kind of screening to identify the cells that have been modified in the way that we want them to be modified. So in terms of editing machinery, there has really been an evolution of different technologies, from zinc finger nucleases to TALENs, and now also to CRISPR and Cas9. So the zinc finger nucleases and TALENs, they work with engineered DNA binding domains, so engineered protein, which is quite a complicated task. And really the revolution with CRISPR-Cas9 is that there the specificity is not conferred by a protein but by an RNA, and that's much more easy to design and to produce. So CRISPR-Cas9 uses an RNA guiding nuclease. So the nuclease is guided by the RNA, and this allows you, and that's true for all of the 3 methods, the common goal really is to introduce a double-strand break and either remove or replace sequences. So removal will lead to a knockout, and replacement can be used to do a knock-in experiment. So just a quick word about CRISPR-Cas9 for those of you who are not familiar with it. CRISPR-Cas9 is originally a self-defense mechanism that was found in bacteria and then was modified to become a genome editing tools. So when we use CRISPR-Cas9 for genome editing, we basically need 2 components. we need the Cas9 endonuclease, and we need this SG or single guide RNA. So this single guide RNA is engineered with a targeting sequence that will bind to your target sequence, and also other domains that will bind to the Cas9, like a nuclease recruiting sequence. And both together, they will form a complex that we call the Cas9 sgRNA ribonucleoprotein complex, or just RNP complex. So the sgRNA serves to direct Cas9 to the specific site in the genome. It's about 20 base pair sequence that will be recognized, and once the Cas9 finds the DNA sequence that matches the guide RNA, the two nuclease domains here, they will cut and introduce a double-strand break. So double-strand breaks are quite heavy for the cells to repair, so what they will do in most cases is that they do non-homologous end joining, and this is error-prone, and that's exactly what we want. We want to either insert or delete bases. So we call these indels, insertion deletions, and they will lead to a gene knockout. Now, as I said, we can also do knock-in with this technology by providing the cell at the same time with a repair template. So they will use the repair template to repair the double-strand break, and this will lead to an insertion. And in this template, you can introduce whatever you want to do. But it's a bit more tricky to do than a knockout. A knockout is much more efficient. So when using CRISPR-Cas9 for gene editing, the first step is really to design this single guide RNA. We want to have maximum target activity, and we want especially to minimize the target effects. So we recommend to use the available online tools to design the RNA. It's important to design multiple sgRNAs. You can also test the efficiency in vitro before you go into a cell transduction, and we have specific kits that can help you with that, like the guided in vitro efficiency check kit. The goal really is to effectively and accurately change a specific genetic sequence in a cell. So that's really the main aim. Now for the next step, the delivery into the cells. So normally what you want to do is you want to minimize the toxicity of the editing. You want to have a minimum of off-targets effect and high efficiency. So this is true for all cell types, but for stem cells we have additional challenges. So stem cells are quite sensitive to toxicity, so it's important to keep the toxicity very low. And the cells also have to stay, they have to continue to proliferate, and especially they need to stay pluripotent. So anything that can disrupt them in the pluripotency will lead to unwanted differentiation, and this is something we don't want. We want to differentiate later, once they are edited into a specific cell type, but not during the editing. And it's very important to preserve the pluripotency during the editing. Now for the delivery, we have used two methods, electroporation and vesicles. So just real quickly talk about these two technologies. One of the classical methods of doing CRISPR-Cas9 is using plasmids. The problem with plasmid transfection is that in the target cell you will have quite a prolonged expression of the Cas9 and sgRNA, and this is something that can lead again to off-target effects. If the cells are exposed too long to the editing machinery, they will start doing some modifications that you didn't intend. So with both electroporation and vesicles, you don't actually work with DNA. You work directly with the Cas9 sgRNA complex, so the RNP complex. You directly deliver the RNP, and this has really great advantages for avoiding off-target effects. So electroporation is quite a straightforward method. We have two machines here that are commonly used for electroporating hiPS cells. So the complex is formed first in vitro and directly electroporated. The Jessical system is much more innovative system. So Jessicals are cell-derived nanovesicles, and these nanovesicles, they are basically able to carry any cargo. So if we do CRISPR-Cas9, we can fill them with the Cas9 nuclease and the sgRNA in a producer cell line together with some proteins that will help to enhance the formation of these vesicles. You can sequester them on the surface, and they will be in the supernatant of the producer cell line where they can be harvested and then put onto the target cell line, which would be the iPS cell line in our case. And they will, after fusion, they will release the cargo. And again, the cargo is DNA free, so it's just the Cas9 and the single guide RNA. And that's exactly what we want. We want them to do their job in the cell and then disappear, not have any prolonged expression. This really can decrease the off-target effects by a great margin. Now, next step. Now we have modified the cells, but once you have done that, what you really want to do is isolate and then expand the edited cells. So why is that? You don't really want to work with a pool, because now you have a pool of cells. Some of them are modified, some are not. Some of them will have a modification in a completely different locus than what you intended. And also this non-homologous end joining will create many different kinds of insertions and deletions, and you might want to have a bit more control over which insertion and deletion you will work with in the future. So it's really a good idea to isolate all of these clones and then pick the one that is really perfect for your application. So for single cell cloning, for doing that, what you really need is you need cells. You need to isolate cells that can later form a colony from this single cell, and that's really challenging for stem cells. You want, of course, to be able to work with this colony so that it can be passaged. You cannot just work with a few cells. You will want to have more cells, and during all the time you want the cells to maintain the stemness and also a normal karyotype. So for this part, for the single cell cloning, the DevCS really is very good, and I show you some data here. What we did here is just to perform the single cell cloning part, and we compared DevCS with two competitor media. We used here 3 cell lines, gypsy 12, 18, and 22. Different cell lines, they behave differently. They have a varying robustness. So for us, for example, C22 is a very robust cell line. C18 and C12 are somewhere in between in terms of robustness. So it's good to test different cell lines because they don't behave the same way. We cultured each of these cell lines in the specific media for 3 weeks to adjust them, and then what we did is limited dilution. So in limited dilution, you make a serial dilution of the cells down to dilution that gives you on average 1 cell per well. We then grow them for several days and observe them under the microscope, and we count the number of colonies that were able to form. So that's really the first step, that you have cells that survive the single cell step and then can form colonies. And you can see here in red with the DevCS, with all 3 cell lines, we got a very high proportion of wells that were able to form a colony, so a high efficiency. So these are the cells that you can then continue to work with. We mentioned before that it's also very important that the cells not only grow, but that they remain stem cells. So here you can see a characterization of the cells. We looked again at pluripotency markers TRA160 and SSEA4. You can see the number of positive cells. Gypsy 12, Gypsy 18 and Gypsy 22 in DevCS, you see that all of them have very high levels of expression of pluripotency markers, indicating that these cells are still stem cells, of course. So this is paramount in order to be able to continue working with them, because if they already differentiate during the single cell cloning step, you will have a lot of trouble downstream. So if you combine this data, the cloning efficiency with the FCS is about 28%. So this gives you a lot of clones to work with. If you are working with a plate, you will have a lot of wells and you will have several clones to choose from later. Okay, so last step, now we have single cell clones, basically, clonal lines, and now it's time to characterize these lines. So we can screen them, you can identify which cells are correctly edited. This really depends on the modification that you have performed. And it can be very simple. If you have a fluorescent marker, for example, it can just be a fax that you can do here, but sometimes it needs more intensive techniques like sequencing to really see what has happened in each of the individual clones. And here we can really help you with our guided product line. We have several kits that help to sequence indels and check the genotype of your clones. So this is the general overview, and now I want to show you an actual example of what we have done in our labs with the FCS. So we will show the complete workflow again, not the design part, because as I said, this is something that you can do with online tools, but then for the delivery, we used both of the two methods that I showed you, vesicles and electroporation. Then we do the single cell cloning using the FCS because we just saw that it's very good for supporting the cells during this process. And then we also show you the screening results. So here's the general workflow of what we did. Our aim was to create a clonal line that is deficient in CD81. So we want to knock out CD81. CD81 is a membrane protein that is found in many cell types. So we chose this because it's also very easy to detect the loss of CD81 using a FACS. We used two cell lines, GYPSI18 or GYPSI22. So again, it's good, at least here for this kind of experiment, to use several cell lines because not all of them behave the same way. So we used both electroporation and vesicles directly with the RNP complex. 1 week later, we make a FACS analysis for pluripotency first, so just to check that they are still pluripotent, but also for the percentage of the knockout that we have achieved. So here in this scheme, the red cells represent knocked out cells. We also, 1 week later, did another FACS sorting. So we sorted the population of CD81 negative cells and then seeded them using FACS or limited dilution into a 96-well plate to isolate, because this here is enriched for CD81 negative cells, but they still represent a pool. So here you will have cells that have different modifications that led to the loss of CD81. But what you want in the end is a clonal line, so you have to separate those. So we do this using FACS or serial dilution into 96-well plates in the FCS, and then we expand all of the ones that are expandable into a bigger plate with bigger wells, and we end up with a clonal population that will have again the self-renewal capacity of stem cells. So here I show you the data that we got from that, from the first step. So here it's a genome editing using CRISPR-Cas9, and the result of that you can see down here. For the two cell lines, we were able to get CD81 knockout compared to the negative control. So we had more than 80% knockout efficiency in the two cell lines, while at the same time still keeping the pluripotency of the cells, as you can see here with its two pluripotency markers, OCT4 and SSEA4. So I'm just going to show you also the data behind this, because this is FACS data. You can see here first the knockout. On the left you have unlabeled cells, so that's negative control. This is negative staining. Then we have the negative control of the CRISPR-Cas9, so these are unedited cells. but labeled for CD81. We can see here a population of cells that are positive for CD81. So that's the wild type cells. And then once we do the editing, you can see a very clear shift of the signal towards negative. So all of this population here, they are CD81 negative, so they have been successfully knocked out. So these are the cells that we will continue to work with. Then also for the pluripotency, we have here the FACS plots, unlabeled cells as a control, and then the two markers OCT4 PE labeled and SSEA4 PE labeled. Negative control, so that's unedited cells. You see that they are stem cells because that's what we start out with. That's our starting point. After editing, you see they are still stem cells. They still have the same OCT4 signal. Same for SSEA4, positive for this marker before the experiment, and after the knockout, they are still positive for SSEA4. So we are confident that these cells are still stem cells. So now the next phase is, now that you have these edited cells, to create the clonal lines. And as I said, we also include a step of sorting the population. So we have this luxury because CD81 is a surface protein. We can easily label it. So we can enrich for only the edited cells. So depending on what you are doing, this might not always be possible to do this kind of enrichment before. So it's kind of an optional step, but it can really help if you can do it to enrich for the positive cells. So here is what we did. We see here staining for CD81. FITC labeled. Here are the cells that are negative for CD81. So these are the correctly knocked out cells, the CD81 knockouts, and these are the ones that we gated so that we chose for the sorting. And yeah, here you can just see a control plot again for the SSEA4, so they are all still stem cells. So we took these cells and continued only with this population and not with the unedited cells. So now they are sorted, but they are still a pool of cells because, as I explained before, they might have different modifications. Some of them might not be the correct modifications that we want. So what we want is clonal lines, and in the following step, we isolate the single cells from the sorted population, seed it into a well to obtain the colonies. So we use two technologies, and it really depends on what kind of equipment you have. A FACS sorter is of course very convenient because you can tell it to put only one cell per well. If you don't have this kind of capability in your lab, you can also do the limiting dilution, where you dilute the cells down to a level where you only have a single cell per well on average. So both methods can be used, have both advantages and disadvantages. So we did this with the two cell lines. So these are now all CD81 negative cells that we sorted this way using FACS and limiting dilutions to compare. And then we looked at the number of wells that had a single cell. So that's the ones we can work with, 5243. For the chip C22, unfortunately, we had a problem with our FACS machine, but we still had the limited dilution that we could work with. So as I said, both methods have advantages and disadvantages. FACS needs a lot of equipment, a bit of know-how as well. It's a bit more rough to the cells compared to limited dilutions, but yeah, it's up to you to choose the method that you're most comfortable with. You also get cells that have two clones in the well, so these you cannot use because you would end up with a double clone, so to speak, and as we said before, what we want to have is a single clone, of course. At this stage, you can start to scale up to kind of a bigger well size, so you can go to a 48 well plate, for example, to continue and to expand these colonies further until you can characterize them, because you cannot really characterize when you have only 10 or 100 cells. At this stage, you can also look at the cells, of course, under a microscope. So this is what I show you here. You can see that we have cells that look very healthy, so they look typical for culturing in DEF-CS. They are taken 2 weeks after seeding. So all of these cells that you see here, they are clones from a single cell that has grown to form a colony. So that's exactly what you want. You want them to be all identical. Now some of those clones we have then further characterized. So we see in the table we have a lot of them. So it's no use to characterize all of them. You don't need 50 characterizations. So we just picked a few of them and looked both at the knockout, just to make sure that they are still knocked out, and also pluripotency again. So both the limiting dilution and the FACS signal cells sorted cells had the knockout, you see, except for one line actually, line number 4. So this clone, we also analyzed it further and we saw that it was heterozygous. So it had one wild type allele that still partially expressed CD81 and one mutant allele that was knocked out. So that's also why it is important to do clonal selection, because if you would work in a pool, you had a mix of cells that don't express CD81 and some have still residual expression of CD81. And it's also why it's important to work with different clones, with several clones until the end of the characterization, because now we can eliminate, of course, this clone, because that's not what we wanted. We wanted to knock out CD81 completely. So here we can see that for one of these clones, this is not the case. And of course, we also looked again at pluripotency. So these are here in purple, green and red, OCT4, TRA160 and SSEA4. Another important point that I mentioned quickly in the introduction is karyotype. So when you stress stem cells or when you culture them for several passages, you always have a risk of karyotype instability. So we have some chromosomal abnormalities, and genome editing is a stress to the cell. You just saw all the steps that are necessary. It's important to have a culture system that will keep the karyotype normal. So we took 5 of the clones that I just showed you in the previous slide and looked at the karyotype that they have and just assured that of the 5 passages they are still normal karyotype. And it was the case. I'm not an expert on this karyotyping, but these all have as a result a normal karyotype. So that's a very important point, because you don't want to have this chromosomal abnormalities. You will not be able to get rid of them, and you will have them in your downstream application, and it will greatly influence the readout of your experiments. You want to have healthy, positively edited cells. That's why it's important to have a culture system that can really keep the karyotype stable, as it is the case for the DEFCS. So now that we are sure that we have several clones that are healthy, they are CD81 knockout, they are still stem cells, they can expand and grow, we are almost done. The last step is the characterization. So characterization here means to really look at what has happened in the cell, and that's a very interesting characterization to do. Of course, you want to know what has caused the CD81 loss, and that's what we did here with sequencing. So we have clone 3, 5, 6, 11, and 9. You can see the 2 alleles and what really happened. So here, for example, in clone number 3, we lost 1 nucleotide in one allele and 7 nucleotides in the second allele. And this has led to loss of CD81 expression. In 5, for example, we had a bigger deletion in one allele and a small insertion in another allele, which will create a frame shift and also loss of CD81 expression. So you can see here different scenarios that can happen in the cell. It's all very random. So it's up to you then to choose the cell or the clone that is most appropriate for your experiment. You can see here in this table just a summary of all the indels that we have detected. We also had one that had a very big insertion here of several nucleotides on both alleles. So also this led to CD81 disruption due to frame shifting, but compared to the deletions, it's of course a much more rare event, and you would probably not choose this clone here to go on because it has a lot of additional bases. So here you can really see the end result. So now you have a clonal line that is well characterized, that is knocked out for CD81. You still have the cell line that is unedited, so you can use this as a positive or as a wild type control, and it's isogenic line, so it has exactly the same genetic background except for the mutation that you have just introduced and also analyzed. So this is really the end of the experiment, and then you can take it to your downstream application. So in conclusion, we have shown here that it's possible to efficiently gene edit stem cells using CRISPR-Cas9 and the DefCS system. It's important to avoid any off-target effects, and one way of doing that is working directly with RNP, with the complex itself and not with plasmids, and that's possible using electroporation or our Jessical system. It's important to have survival of the edited single cells. It's important that we can expand those single cells in the end, and here it's really where DefCS can help. It can very well maintain pluripotency throughout the whole experiment, and it also assures a normal karyotype in the majority of cases. So DefCS is really an ideal system for culturing the human pluripotent stem cells before and especially also during the genome editing, and also after, of course. So you can use DefCS during the whole workflow. It will really support the cells at each stage. Now, if you have your own hiPS cells that you want to genome edit, we will also launch in the next few days new products that can help you with that. So these two first kits, they allow you to do exactly what we just showed, but of course with your own gene of interest, with your own target gene. So they are the Cellatis iPSC Cas9 electroporation and single cell cloning system. So you can do electroporation and then do the isolation of single cells with the DefCS that is included in the kit. The second system is if you want to use the Jessical method. So if you don't have an electroporator, for example, you can use the Jessicals for that. If you have already a working CRISPR-Cas9 system and just want to do the single cell cloning, we also have a separate kit for that. And for the maintenance of the cells before and after the genome editing experiment, we have also just the culture system as a standalone product. So this is for maintenance, and this one is specifically for the single cell cloning because that's where the cells are stressed the most. Then I just want to show you two links that are very useful if you want to have further information. So one is the so-called stem cell tools workflow that gives you an overview of all the stem cell products that Takara is offering. And the second one is also very interesting with all the gene editing tools workflow. So today I talked a little bit about in vitro testing of single guide RNA. It's very important to do that before you even start your genome editing. So we have a guided kit for that. Also the indel identification, so when you characterize your cells, we have a specific kit for that, and the Jessical kit. So this kind of workflow shows you exactly which kit is most applicable to which step in your experiment. Okay, so this brings me to the end of this webinar. So now it's time for questions. If you have any questions, please put them in the questions box and I will try to answer them. Okay, so the first question is whether DefCS includes the growth factors as well. So the answer to that is yes. It contains 3 growth factors that are necessary. So it's really a complete system. You don't have to add anything in addition. A second question is, what is the preferred delivery method from electroporation and Jessical-based deliveries? So this is a good question. Electroporation is good when you are already experienced with electroporating cells. It's a bit harsher method than Jessicals, of course, because you are shocking the cells. But if you already have the machinery, then this is a very good method to use. The Jessicals is more for people who don't have electroporation equipment. It might also work in more cell types, so in more cell lines, in different cell lines, so it really depends also on the kind of cells that you are using. So the choice is really yours. If you have the equipment already for electroporation, you might want to start with the electroporation kit. If you don't, then the chesicle really doesn't need any additional machinery, so you can do it completely on the bench, so to speak. Another question is, how can you check if there was any off-target editing in your cells? So this is not easy to do because you don't know what the off-target effect was. So to be really sure, you would have to sequence the whole genome, which of course is not always economical. So the best way to do it is to characterize several lines and also look at the phenotype of the cells, maybe at the gene expression of some markers that you're interested in, and then probably also to continue working with several clones just to be sure that you don't have any unwanted effect. But it's not an easy thing to do, for sure. Okay, then yeah, another question, but it's again the same question about chesicles or electroporation. So I really cannot answer this definitively. It really depends on what kind of equipment you have, whether you use one or the other. So in our hands, we have run both parallel and both work very well, electroporation and the chesicles. Okay, so if there are no further questions, then I thank you very much for having joined this webinar. If you have any questions, I will write you in the chat box our email address, which I forgot to put on the presentation. So you can always contact us with this address if you have any additional inquiries, or if you want to test these new systems, or if you want us to have a look at your application that you want to do. So don't hesitate to contact us, and I thank you very much for joining today. I hope it has been useful for you. Thank you and have a good day. Bye-bye.